हॅलो फ्रेंड्स, काल आपण सेल डिव्हिजन या चॅप्टर मध्ये सेल सायकल हा पॉइंट अभ्यासला. सेल सायकल मध्ये आपण बघितलं एक डिवायडिंग सेल त्याच्या लाईफ मध्ये आढळून येणाऱ्या इव्हेंट्स किंवा घटना ह्या कशा एका क्रमाने घडतात, त्याला आपण सेल सायकल असं म्हणतो. आणि मग हे सेल सायकलचा जो ड्युरेशन असतो तो प्रत्येक ऑर्गॅनिझम मध्ये वेगळा वेगळा असू शकतो. हायर प्लांट्स मध्ये, ॲनिमल्स मध्ये हा लाईफ सायकलचा ड्युरेशन हा 12 किंवा 24 आवर्सचा असू शकतो. आणि ईस्ट किंवा बॅक्टेरियामध्ये इस्टेशिया कोलाई या बॅक्टेरियामध्ये नियर अबाउट 20 to 30 minutes are required to complete the one cell cycle, and in yeast 90 minutes are required to complete the cycle. This is the variations in the cell cycle duration in a different organisms. Now, what is meant by cell cycle? A sequential events takes place in the life of dividing cell. Now, this cell cycle consists of two main phases. First is interphase and second is M phase. This interphase actually, someone's thinking that this interphase is a resting phase, but actually this is not a resting phase. During that time cell divides itself. During that time cell prepare itself for division. So cell is a very active, metabolically active during interphase, and this interphase occupy the large portion of that cell cycle. Means 95% part of that cell cycle occupy the interphase. This interphase is further divided into three stages, that is the G1 phase, S phase and G2 phase. Now during G1 means during that interphase cell growth takes place, and cells nuclear content is also double. Means that the cell that is G1 phase and G2 phase, cell prepare itself, cells growth takes place. Means a cell organelles proliferation takes place, RNA synthesis takes place, protein synthesis takes place. That is will be takes place during G1 and G2 phase. And in the S phase, actually most important event takes place during the S phase, that is the synthesis of replication of DNA and synthesis of a histone proteins which is required for organization of a chromosomes. That are the most important events takes place during the S phase. And when that cell transfer from interphase to into the M phase, that M phase is the actual dividing phase. Now today we will start here, that is the M phase. Or it is also called as, this M phase is also called as period of division. Period of division. Now, this interphase during the S phase, that is the DNA replication takes place, so the nuclear size is also increases, and during that time in the cytoplasm, that centrosome division is also takes place in the cytoplasm. This will be keep in mind that in the during M phase, now here actual cell start to divide. M stands for mitosis or meiosis. आता M म्हणजे काय आहे, एक तर मायटोसिस असेल किंवा मिओसिस असेल, त्याला म्हणायचं M stands for mitosis or meiosis. And during that M phase, two most important events are takes place. These are karyokinesis and cytokinesis. These two important events takes place during this M phase, that is the karyokinesis and cytokinesis. Now during karyokinesis, what happens? A nuclear division takes place. Nuclear division takes place and formation of two daughter nuclei. Formation of two daughter nuclei. This is about the karyokinesis. Now, what happened during karyokinesis? Sorry, cytokinesis. Division of cytoplasm and two daughter cells are formed. Two daughter cells are formed. Means this is that will takes place during the M phase. M phase मध्ये काय काय होईल? M means एक तर मायटोसिस असेल किंवा मिओसिस डिव्हिजन असेल आणि ह्या मायटोसिस मध्ये पण आणि मिओसिस मध्ये पण दोन महत्त्वाच्या गोष्टी होतील त्या म्हणजे कॅरियोकायनेसिस होईल आणि दुसरं सायटोकायनेसिस होईल. This is about the M phase during M phase and now here the cell cycle is completed. Then next we will start now here types of cell division. Types of cell division. In animal cells there are three kinds of cell division. Three kinds of cell division. There are three kinds of cell division in animal cell. First is mitosis. This is a direct division. Second, mitosis. This is a indirect division. And meiosis is a reductional division. तर अशा प्रकारे सेल डिव्हिजन या चॅप्टर मध्ये आपण एनिमल सेल्स मध्ये आढळून येणाऱ्या या तीन प्रकारच्या डिव्हिजन्स अभ्यासणार आहोत. त्याच्यामध्ये वन बाय वन आपण जाणार आहोत. मायटोसिस अगोदर बघूया, नंतर मायटोसिस आणि नंतर मिओसिस आणि या ठिकाणी ते चॅप्टर संपेल. हा छोटासा चॅप्टर आहे पण अत्यंत इम्पॉर्टंट असा हा चॅप्टर आहे. आपण 12वी मध्ये नेहमी आपण म्हणणार की 11वी मध्ये आपण सेल डिव्हिजन हा चॅप्टर शिकलेलो आहोत. आपल्याला परत हे सगळं सांगत बसायला तेव्हा तिथं वेळ नसतो. त्यामुळे आपल्याला आत्ताच हा चॅप्टर चांगल्या पद्धतीने समजून घ्यायचा आहे. लक्ष द्या. बघूया आपण मायटोसिस इज ए डायरेक्ट डिव्हिजन. मायटोसिस मध्ये काय होतं बघा. आता याच्यामध्ये बघितलं आपण मायटोसिस हे डिव्हिजन हे युनिसेल्युलर ऑर्गॅनिझम मध्ये आढळून येणारे डिव्हिजन आहे. एक्झाम्पल्स आहेत युनिसेल्युलर ऑर्गॅनिझम्स, त्याच्यानंतर ॲबनॉर्मल सेल्स. आता युनिसेल्युलर ऑर्गॅनिझम्स मध्ये आपण एक एक्झाम्पल घेऊया, पॅरामेसियम. देन पॉइंटर मेम्ब्रेन सेल्स, then endoderm cells, That is the examples of a mitosis always shows a mitosis type of a division, and due to that this type of a division takes place, what happen? Structural and functional irregularities arises. रचनात्मक आणि कार्यात्मक irregularities येतात organisms मध्ये. बघूया what happened during mitosis. A meganucleus, you know that in paramecium consists of two types of nucleus. One is a micronucleus and one is a macronucleus. Micro means small, macro means large. असे दोन प्रकारचे न्यूक्लियस असतात. त्यामध्ये मॅक्रोन्यूक्लियस मध्ये डिव्हिजन होते. आताच आपण बघितलं डिव्हिजन होते म्हणजे काय होतं? कॅरिओकायनेसिस होतं आणि दुसरं काय होतं? सायटोकायनेसिस. These are the two important events takes place during cell division. Now, here suppose I will give here a example. Look here, this is a nucleus, and first what happened? Nuclear division takes place first, and then cytoplasm division takes place, followed by karyokinesis. Now, what happened? Look here, this is the nucleus. This nucleus first elongates, enlarge. This is the nucleus elongates, enlarge, and it shows a constrictions in the nucleus, and then this constriction goes deeper from this side, goes deeper from this side, from both the sides. Constriction goes deeper, and this now two nuclei are formed from a one nucleus. Two nuclei formed from one nucleus. This is called karyokinesis. This event takes place first. Then now, what is meant by cytokinesis? A division of cytoplasm. Like this way, cytoplasm divide, and finally we get two cells containing one nucleus. One nucleus goes to in this cell, one nucleus goes to in this cell, and finally we get a two cells, two new organisms. This will be takes place in the lower organisms like unicellular organism, paramecium, and abnormal cells, Pointer cells, and in endoderm cells. This type of cell division occurs. That is a mitosis, or it is a direct type of division. Directly nucleus divide and then cytoplasm divide. And here nucleus remains as it is. It will be not disappear, reappear. Like in mitosis we will see that is the nuclear membrane disappear, later on appear. This will be not take place here. Nuclear membrane remains as it is. Nuclear membrane is not. Nuclear membrane retain as it is राहतं. तसं म्हणजे निघून जातं, परत वापस येतं न्यूक्लियर मेम्ब्रेन. असं आपण मायटोसिस मध्ये पुढे ऐकणार आहे. तसं या ठिकाणी होत नाही. न्यूक्लियर मेम्ब्रेन रिमेन किंवा रिटेन म्हणजे तसंच राहतं. दिस इज अबाउट द मायटोसिस. फर्स्ट व्हाट हॅपन? karyokinesis, division of nucleus, and then cytokinesis, division of cytoplasm. This is about the mitosis. Thank you.